Thank you, Kira, for the nice introduction. And hello, everyone. And thank you so much for the invitation. I'm truly happy to be here because I finally have the chance to share a bit of what we have been doing over the past year in our Berlin Datum research group and how the Hustedt Collection has completely shaken up the quiet rhythm our, of our scientific lives here in Berlin. So let me take you on a short journey through the past, the present, and the future of the Hustedt Collection. Before I take you into the details, let me give you a brief overview of our journey today. I will start from the roots of the collection a century ago through its transformation and new home in Berlin. And then I go to the digital and collaborative future we are now building. Let me start with why the Hustedt Collection matters. Today, the Hustedt Collection is probably one of the largest, historically most important datum collection in the world, and one of the most complete. I think it as a collection of time capsules of biodiversity. Each slide we have captures a tiny moment of life, a place, a date, or a community of species, something that may no longer exist in the same way today. Inside many of those slides lie the type materials that define datum species names, the very foundation of all taxonomic and ecological research. Taken together, these materials give us an invaluable baseline for understanding how environments and species have changed over the past century in water quality, in climate, in distribution. But beyond their scientific value, these historical collections are also part of our cultural heritage. They tell the story of how generations of researchers explore microscopic diversity, share their discoveries, and slowly build the framework of modern phycology. Now, I would like to tell you the story of this collection and of its remarkable collector. So let me take you back to the early 1920s. Friedrich Hustedt was an art teacher and later the principal of an elementary school in Bremen, Germany, where he taught for 32 years. But in his free time, he devoted himself completely to the tiny organisms that fascinated him, the freshwater datums. He began collecting samples from rivers and lakes across northern Germany, and soon his friends and colleagues were sending him material from all over the world. And what began as a hobby became a life's calling. Through meticulous work, Hustedt built the largest private datum collection of his time. But what made his approach unique was his systematic documentation. For each sample, he kept a detailed record of origin, collector, and publication reference. And that level of organization, rare at that time, allow us today to connect his material precisely to their localities and names. Because of his expertise, he was released from teaching in 1939. He was already 53 years old at that time to dedicate himself entirely to datum research. During his lifetime, he published and described almost 2,000 new taxa, 5 genera, 350 varieties, and 139 forms. But by the 1960s, aware of his legacy, he decided to safeguard it. In 1963, he sold his collection and library to the city of Bremen under one lasting condition. It must remain complete professionally cared for, and accessible to future scientists. 2 years later, in 1965, the Friedrich Hustedt Center for Diatom Science was established in Bremen. It later became part of the Alfred Wegener Institute, where it received institutional support, continued to grow through donations, and became a cornerstone of global diatom taxonomy. But the story didn't stop there. The collection kept growing thanks to the contribution of many distinguished diatomists. Over the years, it received personal collections, project slide sets, and type preparations from researchers around the world. Among the most important additions are the complete slide series from Grete Hasle, including many isotypes, and Kramer's nearly complete slide and sample collection. Over the following decades, a series of devoted curators, Reimer Simonsen, Richard Crawford, and Van Vesteri, expanded, cataloged, and digitized, God, the word's difficult, the collection. They created its first digital database and opened it to researchers worldwide. But like many historical collections, its journey wasn't entirely smooth. After Van Vesteri moved to a university position and Friedrich Hinz, the technician, retired after 38 years of dedicated work for this collection, then the collection was closed for a few years. Then in 2024, last year, after more than 70 years in Bremerhaven, the entire collection was moved to Berlin Botanical Garden and Botanical Museum. It has been a long journey, but a faithful continuation of Hustedt's vision that his collection should always remain alive, growing, and open to science and discovery. So let me tell you a little bit about the scale of the collection. The heart of the collection still comes from Hustedt's own work. He personally prepared more than 60,000 microscope slides and about 20,000 samples of dried or glycerine material. Today, the collection holds over 100,000 slides, many of them type material. In addition, there are more than 50,000 raw samples, sediments, and material collected from every continent, including rare samples from early polar expeditions. And all of this is tied together by a historically rich library, the Hustedt Library, which includes more than 9,000 publications, books, and reprints, many annotated by Hustedt himself. What makes this collection truly unique is its integration. The way slides, samples, and literature are all interconnected and carefully cross-referenced. The true strength of the Hustedt collection lies exactly in this threefold structure. It's not just a group of samples, but an entire scientific system. First, we have the slides, which form the visible face of the collection. Each slide is carefully labeled, often with handwritten notes indicating the locality, the collector, and the publication reference. A level of documentation that remains exceptionally even today. Second, we have the raw samples, the sediments from which the slides were made. These are, in my opinion, the hidden treasure of the collection, allowing us to revisit Hustedt's material with modern analytical tools, scanning, electron microscopy, morphometrics, and potentially even molecular studies if DNA can be retrieved. Some of these old samples still contain undescribed species waiting to be studied. And then third, the library and archive. This part connects the specimens to their intellectual context. The publications, reprints, and handwritten notes, including Hustedt's field books and his correspondence with diatomists around the world. What makes this structure truly unique is that all three components are interconnected. For a single species, you can trace the type slide, locate the corresponding room sample, find the original description, and even read Hustedt's own notes about it. This is a model of documentation that really bridges generations of datum research. Now the funny part, the move to Berlin. After being closed for several years, the collection was finally ready for its next chapter. And last summer in 2024, it was transferred to the Berlin Botanical Garden and Botanical Museum. A move that will ensure its long-term preservation and future accessibility. More than 1,500 small specimen boxes packed inside 300 moving cartons traveled to Berlin under carefully controlled conditions. And this was a major collaborative effort between the Alfred Wegener Institute and the Botanical Museum. But how can I say, there was a problem, in my opinion. During the years the collection was closed, a new research group had moved into the former Hustedt Collection offices in Bremerhaven. Step by step, the material had been carefully packed away, yet without someone who truly knew the internal structure of the collection, much of its original order was lost. Slides, samples, and documents were placed into hundreds of boxes, all meticulously labeled, but not always in ways that reflected the original system. When we received them in Berlin, we soon realized that no functional digital database existed anymore, and that we will have to reconstruct the organization from the physical material itself. What we actually received, when the boxes arrived in Berlin, you can imagine we faced both excitement and challenge. And what we actually received in terms of documentation were 6 enormous Excel spreadsheets and a huge folder of images files. Valuable, yes, but difficult to interpret without a clear structure. The biggest challenge now is to connect everything, to understand which sample, which slides, which images, and which literature reference belong together. The collection now was placed in a climate-stable room in the herbarium, which is actually a bunker. And together we have begun to stabilize and reorganize the material, checking the condition of slides, opening boxes one by one, replacing fragile cover slips, and carefully rebuilding the original order as best as we can, and reusing them in archival trays. Each week, as you can imagine, brings new surprises. A mismatched label, a forgotten locality note, or a hidden link between specimens and one of Hustedt's publication. It's a slow, careful work, but deeply rewarding, like piecing together a vast scientific puzzle. And I love puzzles. So every small connection we restore helps bring the collection back to life. The Hustedt Collection is a vast and complex universe. And working with this century-old collection is been inspiring, but like I say, challenging. First, there is the scale. We are dealing with well over 100,000 slides, tens of thousands of raw samples, and thousands of publications, all created and organized by one person in his own system a century ago. Second, there is the fragility. Many slides are old and delicate, cover slips are cracks easily, and some labels are fading or detaching. And third, there are the data gaps. The original digital database no longer exists. We have, like I said, Excel sheets and a folder of images, which are valuable fragments, but without the internal link that tells us which slide belongs to which sample or publication. And of course, there are limited hands and time. At the moment, the curatorial work is done by my colleague Wolf-Henning Kusberg and myself, with the invaluable help from my predecessor, Regine Jan, who still supports the process once a week. But balancing long-term creation with new research and digitization is demanding. Yet, despite all this, every box we open and every record we clarify brings the collection a little closer to being whole again. And please don't take me wrong, these challenges are not obstacles. They are simply the path forward. And with every small step we take, we make this remarkable collection more accessible to the next generation of researchers. Well, out of this apparent chaos, we are now planning the digital future of the Hustedt Collection. The vision is clear: to build a comprehensive, searchable database that links every slide, sample, and publication so that researchers everywhere can access type material, metadata, and images from their own desk. This will, of course, require a major digitization effort, capturing and standardizing all this information. And to make this possible, we are preparing a dedicated project proposal that will allow us to hire trained staff and implement the technical infrastructure for imaging, data integration, and public access. The concept is already in progress. Together with the bioinformatics team at the Botanic Garden and Botanic Museum, we are designing a workflow to connect the specimens metadata, microscope images, and literature references into a unified database within the museum's collection system. So the next steps is to turn this vision into a reality, to secure the resources, partnerships, and people needed to bring the Hustedt Collection fully back into the digital age. Once the digital foundation is in place, the Hustedt Collection will open an entirely new landscape of possibilities for research. First, in the taxonomy and systematics. I mean, we can re-examine species described by Hustedt with modern tools, like the scanning electron microscope, morphometrics, and, where possible, molecular methods. This will help resolve long-standing taxonomic questions and clarify species concepts that have remained uncertain for decades. Second, the collection provides a unique basis for comparative ecological studies, because we know where and when each sample was collected, we can compare past and present diatom assemblages. For example, to trace changes in water quality or eutrophication or species distribution driven by climate and human activity. Third, there are the biogeography. The collection documents datum diversity from more than 90 countries, making it ideal for studying global and regional patterns of diversity and endemisms through time. But beyond research, the collection is also a platform for training and education, for master and PhD projects, internships, and visiting researchers who wish to study historical and modern materials side by side. And perhaps the most importantly, it's a platform for collaboration. We invite colleagues to work with us, to engage with this material, to compare, to resample or rediscover, probably, reinterpret, and even expand what Hustedt began a century ago. In this way, the Hustedt Collection will not only preserve the past, but actually shape the future in datum research. The Hustedt Collection has always been the result of collaboration, and that same spirit continues today. We are open to partnerships in taxonomy, digitization, and education. Researchers can access the material digitally. We provide LM and SEM images on request, or they can visit the collection in Berlin by appointment. For larger and more complex studies, we welcome collaborative projects where the work and credit are shared. We already have ongoing collaborations in Cuba, in Mexico, Turkey, and across Europe, and we hope to continue expanding this network. Also, in the coming years, we aim to connect more closely with international infrastructures such as Datum, BASE, GBIF, and the International Datum Society to make sure the Hustedt materials are visible, linked, and integrated into a global research system. We also see enormous potential in students and volunteer engagement, integrating citizen science approaches and training young researchers in curation and taxonomy and data management. Our long-term goal is to make the Hustedt Collection a global hub for datum science, a place where historical data, modern methods, and international expertise come together. And to reach that goal, we need partners who can share data sets, images, funding, and know-how. People and institutions ready to help build this digital bridge between the past and the future of datum research. So the next step is to secure project fundings and to establish a small team dedicated to digitization and data integration. So finally, this the story of Hustedt Collection is really the story of continuity, of knowledge carefully handed from one generation to the next. It began with Friedrich Hustedt, personal curiosity 100 years ago, and today it continues in Berlin, where the collection is being reorganized and gradually reopened to the world. The next chapter will bring digitization, collaboration, and discovery, connecting historical slides with modern data and global research networks. Our task now is to make this heritage usable, visible, and meaningful again, and to turn a century-old archive into a living resource for education and research. And that is an open invitation to you, to colleagues, students, and future scientists to join us in keeping this legacy alive and relevant for the next 100 years. And I mean, in the end, that's what our work in museums is about, preserving the past and inspiring the future. So none of that, of course, I have shown you today will be possible without a remarkable network of colleagues and supporters. So I want to acknowledge my colleagues in Botanic Garden and Botanic Museum, the research group of datums, the herbarium curators and staff. And I wish to thank also the friends of the Botanic Garden whose generous supports help sustains my work the past year. And finally, thank you all for your attention, your interest, and for being today to share this story. I will let you with my contacts, and now I'm happy to take any questions, but even more so your suggestions and ideas. Thank you.
Thank you so much, Nélida. That was wonderful to hear about the current status and the safety of the Hustedt collection, that it's being protected so well. People can use the raise your hand tool at the bottom of of the screen to do that, and we'll call on people. Otherwise, if you prefer, you also may type a question into the chat, um, and we can pass that on and have Nélida work on that. I see that Pat, you have raised your hand. Go ahead. You've got your camera on. Turn your audio on, and go ahead and ask your question.
Thanks, Mark. Thanks, Nélida, for a great overview of the collection and the vision for the collection going forward. So the first thing is I want you to speak to these data. So over the last 20 years, we've seen an exponential um, growth in the description of genera and species and diatoms. And over the last 20 years, we've seen millions of dollars invested in the digitization of diatom collections in North America and in Europe and elsewhere. And over the last 20 years, there have been fewer citations of holotypes, of using original collection materials. So how do you reconcile the paradox that we have worked harder and harder to make material available in all forms, and yet the community is using those resources less and less, at least by citation? If you go into the literature and say, who used a holotype from this collection or that collection, that has actually gone down versus 20 years ago. So how do we reconcile our interest to get resources to people, and that people are describing species all over the place, and yet they're not utilizing the resources that we're providing to them digitally?
Oh, this is really complex. But nice to see you again.
Yeah.
When I think about this collection, the first idea that come to my mind is training, 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 and being really reluctant to teach, to show the next generation why is this important. I get always the feeling when I talk about this historical collection that especially the young community is like not really aware why is this so important. And as taxonomists, I always see when I review papers or something like that, that people don't really look at the type materials. So I will see also my job behind this collection to be really how do you, I don't know, how do you say, when you just go nagging and being behind this promoting and talking about the importance. So if I'm doing now this, like, I mean, that's a huge task, the Hustedt collection, but I'm really wearing the jacket, and you will see me every year in every uh, um, conference talking about why these historical samples matter. Because till now we have been working in Berlin with this integrated taxonomy, but now we are writing the next papers when we are use the traditional taxonomy, the molecular taxonomy, and the Hustedt collection. And so it needs to talk to each other, but I think we need to train and to show first why is this important. I don't know. It's just the idea.
Thanks,
Thanks.
Nélida. If others have questions, please raise your hand or go ahead and type it in the chat. Just wanted to I know that you received this collection only in 2024, Nélida, but can you give us some concept of just sort of the best strategy if someone does want to visit the collection,
Yeah.
and what kind of resources you have at the collection for using it in terms of microscopes, that kind of stuff, or housing or anything like that, or funding.
Yeah, yeah. I mean, in the first year, what we did was, of course, open all the boxes and put it safely in place. Still, every time I get, because this collection gets a lot of questions, I get requests almost every week with someone who needs something. And it was really difficult at the beginning because if I get I get asked the question about the slide, I was like, well, where is in these thousands of slides, where I can find these slides? And then someone else asked about the raw material. So at the beginning, I worked with the bioinformatics department in order to fit these 6 big Excel data spreadsheets to try and find out the link some way. So this is just something that helps me to understand a little bit the collection, but it's not long term, of course. It's just in order that it's practicable for me to find things. But the collection is fully open. I mean, all the materials is, like I said, in the bunker, but all the literature is in our offices, in four rooms full of literature. And we have also the capacity, we have the microscope, we have the electron microscope, we have everything you will need. Just at the moment, we decide not to loan any material yet, because like I said, we don't know what we have yet. And I think it will be risky because I found already depleted material and cracked slide covers. And so as long as I don't know what we really have, I cannot start sending material away. But I offer to make slides and to make also preparations for SEM to send the pictures, or the people can visit us and work with us. But it is important for me, actually, since the beginning when the collection came to us, to make clear I will not do the technician work for this collection. I will like to work as scientists with this collection, to work with people, to cooperate, but not just sending and receiving and so on material. So it has to be really active the way we work with this collection. Otherwise, I cannot afford to cover all these bases. And of course, with the new resources we are trying to acquire, I hope to have a technician who can support me with this part of the curation. But right now, there's no one there.
All right, thank you. So reaching out to you is the best way to
Reaching out
set
to
up
me.
a visit, that visit to the collection.
And exactly. And then I will talk with the colleagues and the students and volunteers, and we will answer the questions as soon as possible when we find what you are looking for.
A quick question about, I visited the Hustedt collection and was absolutely tickled that I was able to view Husted's slides on his microscope. And I'm curious, you've shown several pictures of Hustedt, and I've noticed that there's been different microscopes in front of him. Do you
Yes.
folks have Husted's microscopes so that we can look at coordinates or anything like that?
No, that was the thing when we did this contract of the transport with Alfred Wegener Institute. I mean, it was really, really complex. But at the end, they had the microscope in how do you say, exhibition? An exhibition in the entrance hall of the institute. And they think it looks really nice there. So we try to push with that. Probably one day they will give it to us, because for them it's just nice to have.
Yeah.
We have some microscopes, but not the original Hustedt microscope.
Okay. Karthik, welcome. Good to see you, and please ask your question.
Hey,
Excuse me.
thanks.
I'm sorry. Should I close this window and see? I mean, it will be
Sure.
more.
Go ahead.
Okay.
You can do that. Thanks.
So now I can see you better. Oh, did I close it?
Yep.
Yeah.
Yep. Gone.
No. Okay. Great. Sorry. Hi, Karthik.
Hi, Nélida. Thanks for the wonderful presentation and showing us the snapshots of Hustedt Collection. So during your talk, you mentioned about developing some kind of program or like bioinformatics program for the managing the data associated with this. So just a small suggestion: if this is developed and if it can be released as an open source, it will be used by many diatomists across the country, and at some point we can also, everybody can pull this data at one platform. So that would be, it's just a suggestion. I don't know how difficult it to implement, but just a thought.
I don't know either how difficult it will be. I mean, we have a great bioinformatics department, happily about that, in the Botanical Garden, Botanical Museum. And we are preparing this proposal. And they seem to know what we need. So I am just talking a lot about why I would like to have, how it should look. And they make me clear there is a part called digitization and the other, the digitation and digitization, something like that. Something when you put the data and then when you openly open to the world. So, but they seems to know what they are doing, and I'm learning with them. I just know what I would like to have at the end.
Sure. Thank you.
Thank you. Another quick question that came up is, over the course of with the years that you're talking about Hustedt being there, I'm guessing that there was clearly changes in the mounting media he was using for his microscope slides. Is that a factor in their preservation? I mean, does it go from cedar oil or like cedar gum to Hyrax to Styrax, or how is that playing out as you open these boxes?
I really don't. I didn't found anywhere because I probably I will need the technician or the previous technician for that. Actually, I'm trying to contact her because she works 38 years with this collection, but she's not easy to reach, and she's not like happy to keep working with the Hustedt collection. But I'm trying to find out all these details because only if you open the boxes you notice there are several different methods. I don't know if Regina or Henning know more about that, but I'm been really nagging with the previous technician because of course I'm all the time in contact and pushing and bang Veshteri, but he is so busy and he doesn't know either.
No.
So this kind of question I was asking, I would like to know because of course I hadn't had time to go in detail with all these nights, only with those that someone had asked me to take pictures again or something like that. But there are a lot of different, I don't know, mountings ways that I need to learn.
Yeah. Leila, I see you have your hand up. Go ahead and ask your question, please.
Yes, thank you, Nélida. I heard that there is 90 country from where Hustedt sampled. So this is very important. And you said that the distribution at some area now would be different. And so we can have an idea about climatic change, about anthropic effect, etc. So maybe if there is a way to make kind of database with the distribution by country and by extant species and fossil one, it might help either for to understand more the anthropic effect and the climatic change, and for the geologists it would be a great help. And as well, often some species we say cosmopolite, and some specialists think that they cannot, they are more endemic, etc. It would help in all this kind of discussion to use the diatom as proxy in paleo. Thank you very much.
Yeah, thank you, Leila. Yeah, this is an excellent idea. I just make some notes. And what I was thinking about is because right now I'm working with Cuba, and they had stopped working about diatoms for the past 20 years. And I started to establish contact with the previous people who were with the diatoms. And now we are working exactly to making this comparison between the old and the new. And that happens before the International Diatom Symposium. So when I was there in South Africa, I was also talking with Kartik and with the people in South Africa, because we have with Hustedt a huge samples
Yes.
from it's huge, the one from Indian and from South Africa. So I personally will ask the people to talk with me, because of course they keep working with these places, and they keep giving names. They change the names, and I need to keep actualize also the Hustedt collection. But yeah, I will go directly to the countries and offer Hustedt.
Yeah, thank you.
Sarah, go ahead and ask your question, please.
Thanks, Mark. Thanks, Nélida. And I'm really, thank you for mentioning Friedel Hinz. I really appreciate the decades that she worked on the collection. I had the opportunity to meet her and visit the collection in Bremerhaven, and it made such a strong impression on me. The level of expertise that she had, her knowledge of the collection was incredible. And after that visit, when I was interested in working with Hustedt material, she provided LM images and SEM images of types. And I don't know how possible that is now to have that, to have someone who has that kind of dedication to the collection, who's there over the long term, because working with it is just so invaluable. And yeah, I have great appreciation for Friedel for everything she did over those 38 years, and for all of you now in Berlin working to make it accessible again.
Yeah.
Thank you.
I'm trying right now that she to let me visit her with all my questions and pictures and data in order to learn a little bit from her. But yeah, Sarah, I'm here and I need to learn about the collection, and I just can't tell I will be here the next 20, 25 years. So, and I will take care to prepare someone and to keep this collection alive. It's not my only job, but it's one that made me dance. So, and that's for me important. I need to feel to dance, then I will keep working on that. So, and I will need to learn, and I don't know how to say that in English. In German says you have to grow up in the shoes for someone else. I don't know how to translate that. I mean, the shoes I have to feel are really big.
Yes.
But I have the disposition. I need to learn. I'm a quick learner. So, and you will keep asking, and I will have to resolve these questions. So I will be forced to learn as well.
Wonderful. Hira, you've got a question, please.
So, Nélida, I was just curious, like you mentioned, a lot of samples, like the slides in particular, were damaged. So I just
Not
wonder,
all.
yeah, not all, some of them. So how do you plan to, like, I guess, like save them or like retrieve them, so that you have the data on them that's present? You are able to collect them and then further like digitize them.
You know, the slides I have found out that are cracked or detached, I mean, they are each in these boxes. You can see it in the moment you open that there is damage. Of course, I'm not taking them out and looking. No, the ones I repair right now were just necessary to put an under how to say cover slip and under, how do you use the name of that? Because there's the cover slip and the other on the bottom. The slide exactly. Yeah. Just to put a second slide there. In other cases, what I could do was to place on the microscope to take pictures and look for the original material to prepare these slides again. So it depends of the condition and the slides I found. And I haven't opened all the boxes. I'm pretty sure I will keep open boxes and it will be a particular thing I have to do there. But the ones I have done now, they are okay now.
Great, thank you. I
Well,
think Pat.
I promise
Yeah.
I'm back. Pat, another follow-up question.
Well, let me just say that I think it's a great opportunity for folks to think in a collaborative way. I think Nélida has really opened a really great opportunity for us to maybe even think differently about how the community works with a collection. And I hope that opening those ideas up, bringing those ideas forward would be a great opportunity. And I do want to also say that both Wolf Henning and Regina being there also creates a great opportunity for a broader set of collaborations. So the collection's really great. Nélida is going to be a great curator for it. And then there's this other set of researchers there in diatoms, and also the botanic garden generally is also got just some amazing resources there in terms of people and other things and beyond the Hustedt collection. So I think it's really and it comes to us to think creatively about how those collaborations might happen. So I think it's a super interesting idea. And I think it's a great idea for the community to think about new ways to interface with collections.
Thank you.
Yeah, thank you, Pat. I think that's a point well taken. We've got just a few minutes left. If folks have any final questions, please go ahead and raise your hand or type them in the chat. One thing you mentioned, Nélida, was the Hustedt collection also had some other researchers' collections. You said it had Greta Hosle's, and then you said some more names, and I was writing Greta Hosle down and I missed it. Who else was there?
Kramer.
Oh, Kramer.
I talk about Kramer. Yeah.
Okay.
And they have some, I mean, all the material from Slatko, and I don't know, remind me, Regine, Henning, what other things, what beautiful things, because we had also, before Hustedt came to us, we already have a collection, all the collection from Linda Medlin, from Hookinson, and Hustedt brought some other parts, so we are just completing all this collection. But I don't know, Regine, what else do we have in Berlin? Schmitz.
I don't. Yeah, I know where the boxes are, but I don't know what they say. I'm sorry. I don't. It was so much things to put away, and yeah, it was a great puzzle to get all the boxes and things together. It was like a game, playing a game. We're still playing. Nélida is still playing the game, the puzzle game. What else? Simonsen also left, of course, the stuff. Simonsen, I mean, the most important person in the collection was Simonsen, and I think that Simonsen was the one that trained Friedrich Hinz, and that's why she was such a good. I mean, the collection was so well kept because Friedrich Hinz was there. I think the moment she retired. When did she retire? When was that? Do you remember, Nélida?
I don't know. I also als bank, but no.
We did a Festschrift for her, so we should know. But it should be in the middles of the tens, or maybe 2015 or early 2010 something. After that, of course, the collection kind of it was because there was nobody there anymore that knew the collection so well. I think this was much more difficult, for example, for a bank to do the job and also do his research.
And actually, I get also the feeling that the researchers around the world keep sending material because we have a lot of boxes, boxes labeled and completely closed. So we haven't opened it yet because we still need to go through all these systems. But there will be surprises as well. I don't know what
And
collections.
then Nélida, there's the other, the new collection also with all the SEM stubs. Remember?
From Slatko Levko. Yeah.
But I mean, there's a huge, I mean, many boxes of SEM stubs where we have not quite discovered, or at least maybe you have by now, discovered the logic behind it. So we have to see.
I have only find the Excel list with this, but not really find who is the collector. I'm working on that. I will find out. Like I said, I will. Yeah.
And then sometimes it helps. Sometimes it helps when you know people that had worked there, so that helps sometimes to find things. Okay.
Yeah. Zlatko was already twice here in Berlin. He helped a lot because he knows the collection very well. So
good.
The first thing I find out was with him. But
Mark, there's also Hustedt had a long correspondence with Sovereign and Sovereign's collection. There's Sovereign collection materials there, but there's also, from my own view of the collection, Skortsov material, Fricka material. It's really well beyond the broad legacy of Hustedt. There's all those people that were working at that. Of course, there's also Chinolki material there too. So there's a lot of the people of the same era who there was communication with, and I believe exchange of materials with as well.
Wow.
Yeah.
That's great.
You see, I have a lot of things to learn still.
Well, we're coming up to the top of the hour. I just wanted to say thank you to everyone for joining us today, and especially thank you.
Wait, Mark.
Well,
Tom's got a question.
Oh, Tom, I didn't catch that last time. Tom, have at it.
Okay, thank you, Nélida. In this quest for increased collaboration, I was hoping and want to ask, how do we make happen that we get one of Hustedt's types onto a taxon page here at diatoms.org? And I'm thinking, Friedel did a whole SEM examination of Hustedt's collection from here in the States, here in North Carolina. And I think it would be wonderful if we can get, if we can get some of those taxa up as taxon pages in diatoms.org. So also, if you know the location or whatever happens to Friedel's poster of those, SEM characterizations of that collection, that would be great also.
In the file with all the images, I have a file that belongs to Hustedt and his own work. I have a file from Friedel's, all this SEM that she did. So yeah, I mean, I'm the most interested that we just give all this information everywhere. Just let me find out how to because I have just pictures with numbers and still not be able to link these numbers to the material. I'm working on it. But as soon as I do, yes, of course.
Well, maybe I can come up with a couple of target things and see if that's okay with you.
Of course, yeah.
Okay, thank you.
All right, wonderful. Well, I want to say again, thank you to Nélida for your wonderful presentation and really providing us an update on this, this incredible resource that, we've all known and worked with and opened the blue books and opened I don't know if we we have Little Freddy and Big Freddy here in the States.
Yeah.
Those are the different Hustedt books that we use. And,
okay.
And I promise they're terms of adoration. So, but join us again in 2 weeks. We're going to have a talk from Nick Schulte, who comes from us from Jonah Ventures, checking in with diatoms and eDNA analysis of environmental samples. Other than that, we'll have Nélida's presentation up on our YouTube channel in short order, hopefully about a week. It'll usually take us to get it cleaned up and put up there. and everyone can share it with their colleagues, anyone who wasn't able to join us today. Any final thoughts, Sarah? Or
just thank you to Nélida, and thank you to everyone for joining and having your faces on. It means so much to me to connect with all of you. This is how we do our work, is that we're a diverse community of specialists scattered across the world, and we need each other. So thank you for showing up.